input spectrum for the eigenvector and looked at the error in the retrieved reflectance spectrum and how it impacted the retrieval of other of the components. And it turns out that in this type of an inversion, the phytoplankton retrieval is the most robust of any of the retrieved parameters. And the reason is what? Why do you think that is? Why do we do such a better job retrieving phytoplankton than, say, CDOM absorption or particle backscattering? The shape is so different from everything else, and the shape of the reflectance spectrum is so sensitive to changes in absorption that it does a much better job by least squares. It also turns out that when you have, in the numerator, you have a power function for scattering, and in the denominator, you have an exponential function for CDOM and NAP. You can, the variance in your in the fit transfers between one or the other. So you could come up with more backscattering or less CDOM could give rise to the same reflectance spectrum. And so that's why there's a lot more uncertainty in estimating those constituents. And I guess the choice of the CDOM basis vector induced the largest variability, coefficient of variation in the retrieval of the other pattern. So getting CDOM right, because it can, the shape shows up in both the sort of scattering spectrum or the non-algal particle CDOM absorption is one of the problems in it. So it's harder to estimate CDOM from this type of a model. So the conclusion that we come to is that this type of an approach provides really good estimates of components of IOPs, and the assumptions lie in the spectral shape of your eigenvectors, but that the variations in those eigenvectors show up in the variability in your reflectance spectrum, and so you can do a really good job sort of identifying what those features are and figuring out where your basis vectors are imperfect compared to nature. And so the next step was, what do we do? Well, if we allow for spectral variations in phytoplankton, maybe we could do a better job than just putting the error in there. Okay. Did anyone have questions on that now that we broke in the middle of it? I don't think so. Okay. So I already asked you this question and you answered it. Why phytoplankton is better. Okay, so the next step was that instead of having a single spectrum, you can have multiple spectra. And so here are a variety of spectra that I measured from some waters in South Africa where we had monospecific blooms. So literally, when you measured the absorption spectrum, it was only that. I mean, you could look under the microscope, and when it said it was mesodinium, that's all there was in terms of being there. So you can actually use your discrete measurements, and we size fractionated them so we could separate these groups into these really distinct. And you see that there's really sharp and distinct absorption features associated with these different groups that we had identified. And so instead of taking a mean, we actually used all of them. So this is a place in the Benguela upwelling system off of South Africa. It's got alongshore winds lead to this upwelling, offshore transport and upwelling, and there's just every February the waters are just like so rich, and they have these expansive blooms that are really variable with species. So I was there for 3 or 4 weeks, and it just like every day you'd go out and the water was a different color. It was just like this evolution of species succession to incredible concentrations of phytoplankton. So we were on a boat, and I was down in the galley, and a wave washed over the galley window, and it was greenish water. And then literally 3 hours later, it looked like this. I mean, it's so incredible. And this is just a wave washing over the window. And so from shore, we did a bunch of surveys from shore. So here's a little TSRB buoy. It just floats and measures reflectance. So it's measuring upwell radiance beneath the surface and downwelling irradiance. You have to correct the upward radiance across the surface. But the day-to-day changes in watercolor are pretty astounding. So I did exactly the same inversion that I just showed you, except that instead of having one absorption spectrum, I put in 6. And then tried to find the concentrations of each one of these. And so here's an example of the measured reflectance spectrum and the modeled reflectance spectrum over here. And so I don't know if this is helpful or not. This is wavelength. So this is the blue end to the red end. This is the date. So a spectrum lies along this axis where white means it's high in reflectance and black means it's low. So you can see that we had sort of broad blue reflectance, then it narrowed to a peak, like really, really green, and then it broadened out again, and then it got incredibly bright. And so this is just the time course of reflectance, and this is the fluorescence, the natural fluorescence peaks that were showing up. And so the first day, the measured reflectance spectrum looked like this. The watercolor was this color. It was this beautiful, bright cyan. And the measured is the black line underneath it, which they matched almost perfectly. So that's our pre-bloom condition. The next day, the water turned this color. And again, here's the model. The model and the measured, we had some offset problems with the sensor, so there should have been, it sort of went to 0 at about 7:25, and it was a calibration issue that we couldn't backtrack from the sensor. And so the model sort of overestimates in the infrared. But the rest of the spectrum, it does a pretty good job. And so it's finding some combination of phytoplankton composition to mimic this spectrum. The next day, or a couple days later, the water looked like this. It was like a Cabernet color with these incredible peaks in the reflectance spectra. And those are not Raman. They're not Fraunhofer lines. Those are pigment absorption peaks that are so intense in the reflectance spectra because the concentration's so high. And then this is the Dinophysis bloom, and we know it because all of us got sick, because Dinophysis causes diuretic shellfish poisoning, which, so that was pretty, we didn't have to validate that one under the microscope. And so the question is, we can, if it's fitting so well to these features, can we retrieve the species information? It seems like we can. And so, of course, what you have to do is you have to validate. And so, luckily, my colleague, Grant Pitcher, is a phytoplankton ecophysiologist. So he collected depth profile of samples for cell counts. I took his cell counts and the sizes of those cells, and I computed an absorption cross-section from assuming a constant intercellular absorption spectrum. So I basically modeled N prime. And so from his counts, I derived what would be the absorption coefficient at 440 from all of these different groups that we identified over a function of time. And so we initially had diatoms, and then mesodinium came in, and then there was a little dinoflagellate bloom, the big Dinophysis bloom. They kind of co-varied, as tends to happen. They're both dinoflagellates. And then we had this chlorophyte. But what I want you to notice is the difference in absorption. These are all like 0.4, 0.6, 0.8, 0.04, and 0.06. So the diatoms and the dinoflagellates were there, but they were in pretty low concentrations. And so then I put in my results from the model inversion. So I had the 5 eigenvectors, and I estimated the concentration of each of those using the nonlinear regression, and then compared the absorption output for each one of those phytoplankton species, and that's what you see in the black. And so it's not perfect, but you see that it reflects the pattern of mesodinium. It reflects the pattern of the chlorophyte, Dinophysis, the dinoflagellate, and the diatom. And I think what that to me initially was like, oh my gosh, like I cannot believe that I can resolve this complex, evolving phytoplankton structure in this area. And it wasn't until later that it struck me about the magnitude. That in the presence of 0.4 per meter absorption due to mesodinium, I can resolve variability in diatom composition of an order of magnitude lower concentration and distinguish it from dinoflagellates that are also minor contributors to this. And so what that says is that the differences in the composition of the pigments of these organisms, the pigments are really, really important. And when phytoplankton dominate the optical properties, that all shows up in the reflectance. It shows up in the upwell radiance spectrum. And so for people who study harmful algal blooms, this is really amazing. So the question is, yeah, go ahead.
Do you have an idea why the model couldn't give the last axis spectrum?
No, I don't know. It's the difference between the sort of the general dinoflagellate absorption spectrum and the dinophysis was subtle, but I don't know what happened here. And I don't know whether it has to do with some sort of feedback between these two. It did underestimate the diatom too. Like, I don't know. I have to go back and look at that one. Yeah. So one of the questions is that, if you can resolve diatoms from dinoflagellates with this low absorption spectrum in the presence of this, can you take advantage of this in waters that aren't dominated by other things? Because that seems to be something that's really of interest, is being able to detect diatoms. And so, that's the question, and that's the kind of approaches that people can take is, what are the limits over which this type of resolution and distinguishing between species can occur. So, work to be done. Okay, so the hyperspectral inversion for PFTs is a fun one because it's overconstrained. You have so many more wavelengths than unknowns. Not all of the wavelengths are independent, but because you can resolve shoulders and changes in the slope of the reflectance, it's analogous to doing a derivative of the absorption or the reflectance and being able to identify different features. And so that's what makes it very robust. So that's why people are very excited about PACE, because suddenly we'll be getting hyperspectral reflectance from space. And can we use these approaches? So spectral gradients. And the fact that we have this makes it so that we can get to this pigment-based phytoplankton taxonomy. Blah, blah. Okay. But the thing that I think is really critical is that the absorption spectra for your different PFTs, it depends on how you define your functional types. And those absorption spectra have to be really different, and they have to be really different in the spectral region for which you have a lot of radiance signal. So if they're really super different in the red, there's no red light. If they're really super different in the far blue, but you have a lot of CDOM, you're not going to see them because the CDOM will have absorbed all the radiance. So they have to be really different in the region for which there's light. And that therefore they can have differential impact on the reflectance spectrum. Okay, questions on that? Yeah.
What do you think of multi-wavelength versus hyperspectral?
What do I think about it? I think that multi-wavelength doesn't have as many channels. What do you mean?
For your inversion problem.
Well, there's certainly a limit to the number of eigenvectors you can put in. If you've only got 7 wavelengths, you can only have 7 unknowns, right? So you have to pick them. So some people, things I've tried is combining the CDOM and non-algal particle, so that frees up 1. Putting in a single backscattering spectrum, some average slope, that frees up another. So, but you also have to have wavelengths of reflectance that are in the region that lead to variations between where the pigments are, right? So it depends. It depends on where those channels are.
But your signal to noise, that signal to noise would not, that would not be a problem.
Oh, yeah. Certainly the uncertainty in your retrieval is really dependent upon the signal to noise, right? Whether, the slope of the radiance looks like this or looks like this, that's going to make a big difference, just like it does for band ratios, right? So uncertainty is really a big one. There's no error bars on this. I wasn't calculating uncertainty back then, right? I've learned a lot since then. But those fits were pretty tight. Yeah, Ken?
So PACE now, they say hyperspectral, but they're talking like 40 bands or
nanometer, isn't it?
5 nanometer.
Yeah, 5 nanometer bandwidth.
And part of the reason is that I mean, even for Sentinel-3, the sensor is hyperspectral.
Yeah, you just take it.
They just band it together. And in this case, the idea is that if you wanted to do derivative type analysis, you cannot do unless you have sufficiently narrow, and in our case, it's 5 nanometer.
Mhm.
Okay, so we'll
There's a little bit of overlap with
Mhm.
Jason.
and they'll be downloading all.
Yes, yeah, you do download, and that's in the visible, and then there's slightly less in the UV, and I think until 800.
Mhm.
And
And,
for fluorescence, they're aggregating bands. I mean, there's different ways by which you can.
and the reality is, you don't need hyperspectral. It's just prettier when you graph it. But you need to have enough spectral resolution that you're able to resolve peaks and shoulders. And so I think that is part of the thinking is you can do a lot with an AC9. Like you can deconstruct an AC9 spectrum and pull out features of different phytoplankton and resolve them with 9 wavelengths. But you have a whole lot more. You can be a whole lot lower uncertainty as you have more wavelengths that help resolve this part of the peak and this part of the peak and this part of the peak, right? And so the more wavelengths you have, as long as the signal is high, which is part of the aggregation thing. So.
I mean, I think we say, but I think there will be new science we cannot yet anticipate.
Yep.
Coming from new measurements. I mean, anytime we put an instrument in the water that was different, we learned something about the ocean we didn't know.
Yeah.
So.
But you should be able to anticipate pretty well because you have the surface.
Yeah.
If you can't do it on the surface, but I have the spectrum.
Yeah, but we don't have the IOPs to go with the surface instruments. So currently we can do hyperspectral reflectance all the way to the UV. Right, we can do it in the visible, but yet we don't have the instrument in the water to the hyperspectral. And we can do, say, with the ACS from 430 to 750. Again, we don't have UV for absorption. We don't have
Hyperspectral backs.
UV. So we cannot yet interpret the signal in ways that we can really squeeze everything we can out of it. And this is my
Yeah.
opinion.
Oh, I agree.
So,
But, you know.
And if you want to optimize it all to many different kinds of water, I mean, there's pigment that exists in lake we don't have in the ocean. The AC9 doesn't have that. It's 20 bands.
Right.
And so there's all kinds of things that.
But I'm just saying from the reflectance, there's enough hyperspectral reflectance, hyperflows and stuff now. That you should be able to do these conversions. You can't
Oh.
do it with
But
that.
you cannot
Oh, yeah.
aggregate them to some degree. And we cannot use, I mean, one of the arguments for the UV channels is better separation of CDOM from NAP and from fiber factor. But yet we don't have those data sets to go with it. So, I mean, I think you're right. And when I got to the field, what was it? Basically told me 98% of the probability is driven by chlorophll. And that's true, but there's this 2% that if you're able to extract, if you are, and depending on noise, etc., there is information there.
Mhm.
This is the challenge for us. I don't think we've squeezed everything.
No.
Yet.
Mm-mm. I mean, you can run.
New sensor is you was to pass to the next
Right.
generation. Something exciting and new to.
Right.
And there was the hope, and we still don't know about. I mean, we still don't know about base at all. But if there's a perimeter on board, that's another information that together can provide us a different way to squeeze information particles.
I mean, so.
That's my feeling. I might be.
So you can easily run inversions on 5 nanometer resolution radiometry, and in fact, I've done simulations where I, how many wavelengths do you need, and how wide is the band to be able to resolve things. And so, I mean, that's been done, but it's putting the atmosphere on top of all that, and it's doing that sort of simulation, and
That's the best case.
yeah, that's the best case. And, but then, so we ran some simulations, Jeremy and I ran some simulations for the Arabian Sea, trying to separate out noctiluca, diatoms, and trichodesmium, just 3. And in fact, the trichodesmium are gone ever since noctiluca came in. So really we're down to 2 dominant phytoplankton. And because of the fact that the the atmosphere over the Arabian Sea is so dominated by aerosols, and it's just a nightmare. And so trying to run an inversion with 2 phytoplankton basis vectors from MODIS, we couldn't get concentration. We couldn't say, oh, we have 3 micrograms per liter noctiluca and 1 microgram per liter diatom, because we just couldn't resolve, because the uncertainty is so large in the atmospheric correction. But we could resolve presence-absence. And that was pretty astonishing, actually, for a multi-channel, highly uncertain MODIS reflectance to be able to at least say noctiluca is there or it's not. And so, but that's a really tough situation. Most of the world is not like the Arabian Sea. So, does that answer your question? Yeah. Why is the water red during a red tide, even though we shouldn't call it a red tide? Yeah, everything but the red is being absorbed by the high concentration of phytoplankton. And they're absorbing it in usually so close to the surface that the red light is not absorbed by the water, because you're not looking at a large path length through the water. So there's still plenty of red photons being scattered by the cells on the surface. And so it's not that they have a red pigment, which I have read in textbooks before, but the fact that they are such high concentrations and sitting near the surface and scattering just the red light. So, okay. So I think it's 2, and I think that we should take 5 minutes, and then we're going to do a simulation. I'm going to give you an Excel file and have you do some simulations to try to model things. This is another model where we can get these factors, and you can read the paper if you have questions. There's also, you have the handouts. It's pretty straightforward. It's just a way of reformulating attenuation and backscattering ratio. Just another type of inversion that you guys can look at on your own. So take 5 minutes, and actually, so Niels, is everything loaded up for the lab? So they have access to, if you haven't downloaded it, download everything from Dropbox for this lab. Has everyone done that? Okay, take this 5 minutes and do that. Download it. There's going to be an Excel file. There's going to be a PDF of a PowerPoint, and then there's a folder that's, inversion software, and then there's another folder that's GIOP. So just download that whole thing.